Okay, I think we'll go ahead and get started. I'd like to welcome you all to today's lecture and welcome our simulcast viewers from other parts of the country and around the world. Before I start, I'd like to thank Howard Weider and the Canada Excellence Research Chair for funding this seminar series, and remind you that next week we have Naomi Tagg from UC Santa Barbara. She'll be talking about forest ecohydrology and drought and why geology matters. So please join us next week. But today I'm very happy to introduce Professor Amilcare Porporato. Amilcare is now the Addy Professor of Civil and Environmental Engineering at Duke and holds a joint appointment, or a dual appointment, I guess, in the Nicholas School at Duke. He received his master's and PhD degrees from the Polytechnic of Turin, where he was an assistant professor for some years prior to moving to Duke University about 10 years ago, I think. Really, Amilcare has done some stunning things in ecohydrology, and for that he's been awarded many prizes. He was the Lendoit Visiting Chair at the École Polytechnique Lausanne, EPFL. Some of you know the group there. He was awarded the 2007 Professor Senol Award, the 2010 Earl Brown Outstanding Civil Engineering Faculty Award, and he's the 2011 recipient of the Lagrange Fellowship from the Polytechnic Turin. He was also elected to AGU Fellow in 2012. Amilcare serves on many boards, does a lot for the field. He's an editor of Hydrological Processes, one of our main journals in the field. He's been editor also for Water Resources Research. That term is now ended. And he's a member of the editorial board for Advances in Water Resources and the Hydrological Sciences Journal. And some of the students may know that he wrote the book literally on ecohydrology together with one of his mentors, Ignacio Rodriguez Iturbe, and together they've published many papers and really advanced the field on the quantitative side, particularly in ecohydrology. So it's a great pleasure to have Amilcare with us, who's going to talk about soil moisture dynamics and nutrient cycling. Thanks.
Thank you, Jeff. It's really a pleasure to be here, and a very exciting visit and interactions this morning and this afternoon. So the topic of my talk is how I'm a hydrologist and how hydrologic variability actually impacts through the soil moisture dynamics the carbon and nitrogen, which I call nutrients in general, but so nitrogen and phosphorus and other compounds that are useful for plant growth in the soil, this cycling. And at the same time, I want to give you a little bit also of other side of the coin, which is the stoichiometry control, the quality of these compounds that are organic matter in the soil. So just to move forward this way, yes. The outline of the talk is focusing on the temporal dimension more than the spatial variability of these interesting dynamics, starting with a bit of a background very quickly on the stochastic soil moisture dynamics, this random variability of moisture availability to plants and soil microorganisms, to then move to how these controls, hydrologic controls, take place on the soil carbon and nitrogen cycle. And I'll give a couple of examples here, like some interesting dynamics between microbes and the substrate being organic matter. This another linkage, very interesting, between mineralization and immobilization of nitrogen by microbes, and how that is modulated by hydrologic variability. And finally get to the more chemistry side of this, which is the role of stoichiometry, which is in parallel to the one of hydrologic control on that. And then a bit of a more broader view of where I think the frontier of research is in this area. What are the challenges that my group and some of my colleagues are pursuing, especially now bringing in the spatial dynamics of the issue, and finally some highlights on how we can use this knowledge to better manage our ecosystems, agricultural system, for food production, for correct use, sustainable use of soil, and water resources. So before I have to thank my collaborators, most of this work is done in collaboration with Stefano Manzoni, now a professor at Stockholm University, former student of mine at Duke, Giulia Vico, also in Sweden, Tony Parolari and Xue Feng, my postdoc and student at Duke University, and Eduardo Dali, former student at Duke and now at Monash University, and of course the funding agency, the US Department of Energy, the US National Science Foundation, and the Department of Agriculture. So let's get to the story here. The focus of my research is broadly defined as ecohydrology, which is really the interactions between the soil, the plant, and the atmosphere system. And lately, I'm interested in sustainable management of ecosystem, also, of course, the social aspect. And it's very interesting to me, this system, because it's almost like the prototype of a complex system. It has all the elements that make up a complex system. It has lots of stochastic, meaning random component, large number of degrees of freedom that interact together. And it has nonlinear component. It's not a simple linear system. It has threshold. It has a strongly nonlinear interacting component interacting at different scales. So if you think of this as being the abiotic process, the water and the energy cycle, having a two-way interaction with the biotic processes, the carbon, nitrogen, phosphorus dynamic, the microbial and the vegetation component of it. That's really an interesting coupled system that defines ecohydrology for me. And then a similar two-way interaction, if you add the social aspect, is present, meaning that these processes, water, energy cycle, nutrient cycling, affect, of course, the food production, the wealth of a society, but the scale of our action on the ecosystem is such now that there is actually a feedback on that, and through irrigation, through soil, unfortunately, degradation, and salinization, and other issues. So the framework of my analysis is focusing on the root zone of our ecosystems. There is, depending on the type of ecosystem that you have, there is a typical rooting depth, larger for forest, less for grasslands or prairies, like here. But this is really my control volume where I start my analysis, which is a water balance equation, which is taking stock of how much water there is in this rooting zone and how that is intermittently input by rainfall, and then is lost by processes like evapotranspiration, runoff, percolation, and so on. And if you go to field sites and measure how much water is available to plants or to soil microbes or fungi in this root zone, you will see something that looks like this. This is a very arid place in the southwest United States. It's New Mexico. It's a Sevieta long-term ecological research station. You see that the rainfall input is highly intermittent. It's random both in the arrival times and the amounts. And that propagates as it gets into the soil layer into jumps. This is relative soil moisture in volumetric terms. And you see that once there is a big rainfall event, there is a jump. And then between rainfall events, it's almost like a deterministic decay. And that deterministic decay is more regular, it's highly nonlinear, and it's driven by the soil plant atmosphere processes. So the same story you find in a more humid region, like this is the Duke Forest experimental site, just 12 miles west of Duke University in North Carolina. And of course, it's a more humid place, but this is measurements by Eduardo Dali, my former students, in this pine forest, which was a more comprehensive phase experiment, enriched CO2 experiment. But our hydrologic story is as follows. You have a growing season, intermittent inputs, and the soil moisture averaged over the rooting zone, similar pattern, random jumps, and relatively deterministic nonlinear decays. So there is clearly some similarities, some dynamics that we can exploit to do simple modeling, which clearly has to have a random component. And so much of my research has been devoted to this type of of dynamics. We discussed this morning with the students a couple of papers that dealt with the main equation that describes the water balance in this. This is a simple accounting. This is the rate of change. N is porosity, Zr is the routing depth, as I said. This is the rate of change of the amount of moisture in my control volume here, which is input minus output. Rainfall minus interception, runoff, evapotranspiration, and percolation. Nothing really special. It's a simple equation, you will say. Well, not quite. It's highly nonlinear. Many of these terms are strongly dependent on the variable itself. And it's also with the random input. So that immediately makes it a stochastic nonlinear differential equation, which is much more exciting to study, and allows you to do analysis in terms not of the single realization of S, but looking at the probability distributions, what is the chance of observing a certain soil moisture at a given time in a given moment of the growing season, for example. So that's almost like the background. We discussed this morning at length with some of the graduate students. But the way I want to proceed from here is, okay, give you an example of why we have this nonlinearity, and then just take these dynamics and use it as a driver for soil biogeochemistry, carbon and nitrogen cycling. So, of course, just a quick glance at why we have these nonlinearities. Well, it's because of the complexity of the hydraulics of the soil and the plant, and the biology. If you plot evapotranspiration as a function of soil moisture, you will see like this plateau and then decay. That's a very nonlinear function. There is almost like a threshold here that defines the level of stress of plants. And if you go to percolation, it's the same. It's a highly nonlinear process, of course. The reason is that the soil is a porous matrix, highly nonlinear. The plant xylem, whether it's grass or trees, has lots of interconnected pipes that can cavitate, can reduce their conductivity, and probably even more importantly, if you get to the stomates, plants have these valves, the stomates, at the leaf level that can actually try to control a little bit the water losses to optimize carbon assimilation versus water loss. So all of that adds to this nonlinear behavior of the soil system. But let's get to my focus. This was just a premise, I would say, that now let's get below ground in the soil part. Let's let the plants for a moment alone, and let's focus on for example, this is the sketch of the nitrogen cycle in the soil. We all know it very well, but let me just quickly set the stage for the following discussion. So if I follow nitrogen in the soil and the plant system, I have most of the plants shed leaves and roots, and that creates a litter input of nitrogen into the soil, which becomes soil organic matter. I can partition it into litter, microbes, humus. This is an arbitrary distinction, but it's useful for modeling purposes. There is lots of conceptualization here. But as it gets here, this is in form of organic matter, so it's tightly linked to carbon. And it gets mineralized by microbes. This is the engine of the soil, of this cycle. So there is ammonification, you get ammonium, you get nitrogen, all sorts of mineralization processes. And here it's where it becomes available again, either for microbes to be immobilized again, that will come back later, this arrow, or to be uptaken by plants as nutrients. That's why I'm calling this nutrient. This is how plants get their minerals. But of course, depending on the hydrology, this nitrate can be leached. If there is lots of moisture, it can percolate down leach down with the water, or it can be denitrified. Actually, this is an issue for greenhouse effects. And of course, as you go from here to here, it's mineralized, so it's no longer connected to carbon. And so if you're interested in carbon sequestration and in future effects of climate change, you want to probably maximize the carbon storage in that. Now, what has this to do with hydrology? Well, it's all here. This is a famous graph from a classic book that shows microbial activity, so the activity of this part of the engine, as a function of something that is percent of soil pore volume filled with water, soil moisture. Is my variable S that I had in the previous graph. And so this is dry, and this is very wet. And, well, we know that in deserts, you don't have much decomposition. No? Think of the mummies of Egypt. No? They would not decompose because it was very dry. Well, also for other reasons. But think of very humid places, anoxic condition, very slow decomposition. But put a piece of apple or a leaf in the summer of North Carolina, where it's humid and hot. The decomposition is very fast. And so the microbial activity is at maximum, ammonification and nitrification go at maximum speed, and you see this engine going, this cycle going very fast. So now you see the link, I hope. Think of this axis being this up and down, stochastic up and down of soil moisture that I showed you before, which becomes the accelerator or the brake on that cycle. No? And so it becomes interesting now to couple them together. And of course, the physical reason for that is probably known to most of you. It's that as I fill up my soil with more and more water, processes change. And this resulting activity is actually due to the fact that, of course, the gas transport is reduced as I get more moisture, but I get also more solute transport. It's a bit of a compromise between different processes. If I'm a microbe, I like probably not too much dry because I have stress, but I also don't like too humid because predators move easier and they come to get me, like the grizzly bears for Jeff. And so this is how the microbial activity responds to hydrologic variability. Now, if you want to proceed quantitatively, which is what I want to do to get predictions, is let me write equations for this system. Let me try. There are no first principle ways to proceed, but I can try to conceptualize it. And so the previous system is now made up of compartments. I have a litter, I have a humus, I have a microbial biomass. These are the three boxes that you had before. And this is for the nitrogen cycle. And I have a ammonium and nitrate compartment. And then I need to see all the inputs and outputs, so plants, residues, with their own, well, it depends. The addition is organic matter, so I need to take into account, this is an important quantity, Cn. So this is the carbon to nitrogen ratio of the added residues. This is the carbon to nitrogen ratio of what comes out of the litter, and so on. So each of these is an input or an output. And I do this for nitrogen, and I need to have, of course, this is only mineral, but for this 1, 2, and 3, I need to have a corresponding diagram for carbon, because they're coupled. So we did that in this series of papers. Once you take a stock for each of these compartments, you can write a balance equation, which basically gives you, we'll come to that later, and this also later. These are just applications. But the bottom line is that for each of these compartments, there is a corresponding balance equation. And let's not worry about details here, but so these equations you know already. This is the water balance equation. And the other one is, so this is like the litter. So the rate of change of carbon in the litter is the input, addition of litter, minus how much is decomposed by the microbes. And this is another term that is basically how much of the microbes that die go back into forming like organic substrate. So you want to conserve mass in terms of carbon and in terms of nitrogen. This is nitrate. Nitrate can be gained by mineralization and can be lost by uptake from plants or microbes and leaching, percolation. So you see the philosophy. Don't worry about each of these details, but two things I want to emphasize. One is, where is the hydrology? Well, how do I couple the water and the biogeochemistry? Well, for example, through decomposition. Like, the decomposition function is not really known from first principle, but I can say, look, if I have litter, there will be decomposition. If multiplicative with biomass, I need to have biomass to drive the process. But there needs to be a reaction rate that must be a function, this macroscopic reaction rate, that must be a function of soil moisture. Remember that maximum peak. And if we are in a place like here, where there is cold and warm alternation, when then also temperature is important. I will not have it in my results later, but of course, it's as important as soil moisture. And so you see the coupling now with all of these. And of course, there is nonlinearities among all these variables. So it's a very complicated system, if you want. Another example of the hydrologic control is here, leaching, no? Percolation of nutrients. If I have nitrate, this is the nitrate concentration in my notation. Well, this is a solubility coefficient, but the flux, the presence, the leaching is proportional to the leaching to the percolation of water, which is a term here. So you see the coupling. It becomes this random function going up and down, driving a highly nonlinear coupled system, which is quite neat. Now, we did this analysis, we studied the system, and we were able to show how the hydrologic variability propagates at different scales, and this acts as a nonlinear filter of hydrologic variability into the more recalcitrant, slow-varying dynamics of the different components. But what I would like to do is now focus on some of these parts and guide you through a couple of examples that we may find interesting. So the first is, let's look at this part of the system. You see that this is litter, so it's the amount, is leaves, roots that I add into the system. And this is the rate of, this is how much microbes, fungi, bacteria that I have in the soil perform in the. Well, if you look at it, and if you're a little bit familiar with the mathematical biology literature, this is exactly what you will find as a predator-prey system. This is the prey, and this is the predator. And it's actually prone to show oscillations, which are modulated by hydrology. So that was the analysis the first analysis that we did. I promised that I would have, before that, shown this nonlinear filter action. Let me just show you a numerical simulation before I proceed. This is the result of a numerical simulation of the whole system. So this is soil moisture over many, many growing seasons, just an example. And this is how, oops, what am I doing? This is how the hydrologic variability propagates through litter. The fonts are a little bit screwed up, but this is the litter, carbon in the litter. You see how it really only filters the main variability. This is nitrate, still retains some of the spikiness, but at the same time inherits some of the slow variability from the slower pools. And you can see it from the power spectrum of each series, how soil moisture is more looking like a white noise at these scales, and then a low-pass filtering action. Let's see if I can take this away. Low-pass filtering action in the other more correlated series. Now, of course, these variables are difficult to observe in reality. What you can see is just surrogates of these quantities. Like, for example, you can have an idea of how much microbes are active by studying respiration, which is the emission of CO2 from the soil. So that's something that we did in the Duke Forest, measurements by the same guys I mentioned before, plus Gabi Katul, a colleague in the Nicholas School. So we have soil moisture probes. Where are they? Here. Temperature probes and CO2 probes. And without making a very long story, 2 years of data, and you see, let's just focus on one point here. So this is soil moisture. This is volumetric soil moisture, so I use theta. But just look at the trend. So look at the jumps up and down. Of course, there is a wet season and a drier season, so you'll see that. But if you see carbon concentration in the soil, it's almost like 1-to-1, especially during the the warm season where there is activity. So this is a clear sign of coupling between the two dynamics for me. And if you look at this is surface, if you go at deeper layers, you see that the control becomes less important because the deeper you go, the more stable is soil moisture and it becomes more temperature dominated. So to me, this is just for you to have an indirect experimental evidence of these dynamics taking place. It's not a validation of my model per se. But let me jump back to the example of the predator-prey system that I was mentioning before. So we have this, it's a bit of a theoretical analysis, but I hope you will find it interesting. This is a simple dynamical system, no? Soil organic matter, prey, predator. Decomposition, and dead biomass coming there, respiration going out. You write this system, you forget about the forcing, you just take a constant forcing, you can do a stability analysis. You have a stable point, which is your long-term equilibrium, and in this type of system, you do perturbation analysis. You just modify a little bit the stable point and see how you return to it. Whether you return it as a monotonic, oscillating, or you just have an unstable system. And what we found is that, as we would expect, in some parameters, and these are details that I don't want you to worry about, you can send me emails, I can give you details, but there are some regions of the parameter space in which the approach to equilibrium can be as a stable focus, which means oscillations, dampened oscillation. And this is actually happening let's just focus on a more realistic nonlinear model, just to save a little bit of time. This is dry soil, this is wet soil, and you will see as the microbial activity is modulated by soil moisture, if it is slowed by dry condition or by wet condition, you have a focus. If it is full activity, I just go directly to the equilibrium point. And this could be a strong, important signature of hydrology on the behavior of biogeochemistry. Now, is this true? Well, my student Stefano, student at that time, was very happy when he found data in the literature that confirmed, at least in the lab, this type of behavior. Notice the time scale of a few days of this up and down. No? So more microbial biomass, there will be less substrate, and if you then have less substrate, the microbial biomass will go down. And this happens only in some soil moisture conditions. So that's my first part of the story. Let's go down in the system, and I'll give you the second little story, which is, well, now we are in the biomass and the mineral nitrogen. There is a very interesting dynamic taking place here, which is, okay, nitrogen is being decomposed by microbes, and microbes take what they need, but the rest is mineralized. And at some point, they may need some of this mineral nitrogen to grow back again, especially if you feed them with very little nitrogen and lots of carbon. Like if you take sawdust and you throw it in your garden, for a few, probably months, you will have a period of immobilization if there is nitrogen available, in which it's like almost feeding a person with too much bread and too little ham. No? If you give me just too much bread, I will go in the fridge here and get my ham. Okay, and instead, if I don't have ham in the fridge, I will just starve, because they don't like only bread. That's how the microbes think. They need a balanced diet of carbon and nitrogen. So the other interesting thing is that plants at the same time want to uptake the nitrogen, so there is a competition between these guys and these guys for the same ham, nitrogen. So where is hydrology? Hydrology is actually controlling also this competition, because if you study soil moisture, this is an inverted graph, so you get dry and wet, and you study how these decomposers suffer water stress, it's very different. If you're a fungus, you are very strong, and you can go to very low levels of soil moisture and still be in decent shape. You don't want to be a nematode in a drought. You're the first one to go. So if you're ever given the choice don't just try to be a fungus, okay? But bacteria are somewhere in the middle, and if you are a plant, well, there are different levels of tolerance. Corn is probably here. Some grasses and some shrubs, they get all the way to here. So, make long story short, let's go back to my up and down of soil moisture in the growing season, and let's put some of these thresholds of different tolerance to water stress. Bacteria, which are fast. So when they work very fast. Fungi, which are strong, but they work very slowly. And plants, somewhere in between. So who is winning this battle for uptaking the ham from the fridge, so to speak? Well, it will depend on the statistics of soil moisture. So that's, again, something that hydrology has a strong impact on. Time is running out fast, so let me move on to the other side of the coin, which is, I told you, if you're a biogeochemist, so far you will say, okay, there is role of hydrology, yes, but there is not only hydrology. If you take, let's just focus on this diagram. So this is time to degradation. Imagine that you take a liter bag and you put it in the soil. So you take a bag, porous, and you stuff it with waxes, lignin, cellulose, stuff that is more like sugars and stuff. There are different speeds of decomposition, independently of the activity of the bacteria, simply because waxes and lignin are really hard to chew, and these are much easier to go. So there is that aspect which I'm going to call stoichiometry, is the quality of what you have in your as litter. And how can we put the two things together? So that's my second. Of course, on the other side, there is the temperature or there is the moisture. If you are in the tundra, the decomposition is very slow for the same type of decomposition material. If you're in the middle of the Great Plains in Kansas, it's here. This is a wet tropical forest. See, the speed is completely different. So how can we reconcile these two? So the analysis is, again, brought to its minimal complexity to do some simple math together. And let's say that we want to model these experiments of litter bags that the ecologists and biogeochemists do. So we have a litter cohort, as it is called, a bag full of leaves and roots of different quality. There is carbon and there is nitrogen. This is decomposed. Part of this is respired. This is the engine, the microbial biomass, which decomposes, and if it doesn't grow with balanced bread and ham, carbon and nitrogen, it will try to compensate from the mineralization, immobilization, from the mineral compartment. So this arrow can be mineralized or can be immobilized depending on this growth. The key thing is that the carbon to nitrogen ratio of microbes, because of biology, it's very much constrained towards a magic number of 10. That's the typical ratio. So put this as a constraint, constant carbon to nitrogen, start your bag, stuff with litter, and see within the climate, with decomposition, what happens, how it decomposes, and in case, how it mineralizes. So we'll keep constant microbial biomass, we'll change the quality of the litter. This would be the CN ratio of the litter, which is like what defines my diet. And we will have a constant respiration rate, just for simplicity. So let's write down the simple equations. This is the rate of change in the bag of carbon, which is simply lost. That's the only output of carbon. This is output of nitrogen, okay? So carbon is only lost by respiration. Nitrogen is basically either mineralized or immobilized. Let me call it minus mineralization. If it goes, it's actual mineralization. If this is actually a positive term, it will be understood as immobilization. I could have put a slash. But anyhow. But it's very simple algebra. I'm not going to bother you here. But if you impose the constraint of constant carbon to nitrogen ratio of the biomass, the only way you can satisfy this is that this mineralization immobilization must be linked to decomposition. Otherwise, the growth here will be not with constant carbon-nitrogen ratio. So if you impose that, trust me, but it's easy to show that this is actually proportional to decomposition times stoichiometric coefficient, which is the carbon and nitrogen in the liter, which is the ratio of these two guys, which will change, and this constant, which is the number 10 I mentioned before. So now this is interesting, because it's simple, and what would you do now if I were giving you this, speaking to the students, as an assignment? Oh, you say, oh, it's a simple system of nonlinear of differential equations, but how do I do with decomposition? I go back to the previous slides. I have all this soil moisture, carbon and carbon in the biomass and liter multiplied. It's complicated. This is all the hydrologies here. But what happens if you take the ratio of these two equations? Decomposition goes away. You can simplify it. And you basically have dc dt over dn dt. You simplify the dt's. Mathematicians should not listen to this because it's not really orthodox, but it's okay. And you get dc dn. So you get a differential equation in closed form, which is easy to solve. And this gives you curves that are independent of decomposition and depend on the initial condition of quality of the liter. This is a constant, as I said, and this is basically nitrogen as a function of carbon, normalized. So as simple as it is, we went to big data sets of these liter bags, and this is results of two papers, one in Sciences in 2008, and then a more refined analysis in a few years later. But the story is of course, there is lots of scatter, but if you plot these curves, n as a function of, just inverted, 1 minus c, what you see is that there is a clear pattern depending on the quality of the initial condition. So this is very low, which means lots of ham, very little bread. This is lot of bread, very little ham. And so this is the full bag, and this is the decomposition, immediately reducing nitrogen. So they start eating and decomposing, and the nitrogen goes down and the carbon goes down. But if you have a very high carbon substrate, you see that there is a phase where nitrogen grows. This is the nitrogen going to the fridge, if I may use this analogy, and getting the ham. No, this is called immobilization phase, and you can actually compute analytically what is the critical point of that transition when they got enough immobilization and they can start decomposing. So where is the hydrology gone? Now it's completely disappeared. Well, these are parametric curves in carbon and nitrogen space where time has become a parameter. So now the hydrology is how fast I go along these curves. If I'm in a very dry place, I will go very slowly. If I'm in a very humid place, I will go fast. If I'm in a cold place, I will go slowly. If I'm in a warm place and humid, I will travel then fast towards decomposition. So that covers the other aspect. And we then added also the phosphorus dynamic, and then now you get more possibilities because you can also immobilize phosphorus and nitrogen, but the story is very similar. You can have just 1 extra degrees of freedom. I'm going ahead because I don't have too much time. But so this is hopefully gives you an idea of how hydrology has a strong impact, and together with that also the biogeochemistry, the quality of the liter. Where is my research and going in this direction? Well, it's trying to add the spatial dynamics. There are really neat open questions that certainly I don't know, and in general I would like to see explored. One is that the microscale, no? The soil is actually a very complex system. We're interacting mineral microbes and soil moisture, and the microbes grow with moisture. They actually grow in, they change the hydraulic properties of the soil. How? Well, this is really has not been explored much. Let's go at a little bit larger scale. This is again Sevieta, the place where I showed you before. There are shrubs. There are patterns of fertility under these shrubs where there is much more nutrients than otherwise, and here there are crusts. So lots of special dynamics going on. It's not all uniform, the story that I gave you before. Then there are some interesting special cases, like waves of mushroom, these fairy rings that are waiting to be explored, and certainly there is nice nonlinear dynamics underlying this. But probably even more interesting for us dealing with water resources and biogeochemistry at large scale is what happens at the regional scale. This is a picture of Logan at the end of the winter. Snow, and I'm sure similar situations around here, but where you have different aspects, slopes, radiation inputs, interactions between organic biogeochemistry, moisture, energy cycle. What are the global statistics at the watershed scale? That's I think, largely an open question. And ultimately, I really would like to see these questions solved, at least tackled, if not solved. What if I take a complex landscape with all its fractal structure, mountains and streams, and how can I predict these patterns that are just observed? This is a combination of data. This curve is just conceptual line, it's no theoretical basis, where I plot carbon to nitrogen ratio and carbon to phosphorus ratio, some key stoichiometric ratios. So, and I go from the sites of photosynthesis close to where I capture my carbon, and these ratios are of course very high. And how do I lose carbon? And I consistently change these ratios all the way down to the stream or lakes, which I know many of you care about here because it's a huge issue. But I don't know what this curve is, and I'd like to see it tackled. Has been called the nutrient spiraling from the hill slopes or the mountains down to the streams. If I have still 2 minutes, I just want to close with, I'm in an engineering department. There is a Center for Sustainability here. How can we use these dynamics for managing better our ecosystems? And I mean, of course, we know this is the feedback of how society can interact with the environment. Oh no, of course, hydrology has a strong impact on society. This is a very nice diagram from a relatively old paper of wars and peace. These are wars in Eastern colonial, pre-colonial Africa, and this is levels of a lake. So every time there was good water availability, peace. Every time there was a drought, almost no food, fighting. And of course, now we are so many and impacting so much the system that, as I said before, there is a feedback on actually the resources themselves. So the way I tackled this in my research more recently is think of this, and it also has to do with food security as well. So normally, all the cycles I mentioned before is plants uptaking CO2 through photosynthesis, getting their mineral nitrogen and this biogeochemical cycling, and then of course you have rainfall that acts as a facilitator of this cycle. It's a system out of equilibrium, which works as a thermochemical engine. Open, gets CO2, gets energy, and spits out biomass in terms of high level of chemical free energy. So what are we doing when we manage it? Well, what we are doing when we manage it is take this same engine and stepping on the gas. And doing that, of course, the goal is to get more harvest for our use, energy, food, fiber. To do that, we need to add most likely irrigation, water, and fertilize. And by doing that, we push the system. This is think of it as a thermodynamic potential landscape. So this is equilibrium, this is the natural equilibrium is death. But the normal functioning is here. When we push it to manage it, we want the system to become more efficient, and we push it. And most likely, we push it away from equilibrium. Then the question is, are there cliffs here? Is this, I don't know what this landscape is, but are there potential degraded state? Certainly, I know that as I push it, I reduce biodiversity, I reduce resilience. And I really would like to know this. I'm sure that in the past, this threshold have been reached. Consider what happened in the Dust Bowl in the 30s in the United States, and probably even here. We were discussing this at lunch. Droughts, improper land use, look at this speaks by itself. The emphasis is now, I'm sure here as well, but in the US, is really to understand this critical zone, which is basically the plant, the soil, and the deeper all the way down to the groundwater and the bedrock through these critical zone observatories, which are both observatories and theoretical modeling experiments. We have one related to Duke, which is the Calhoun in South Carolina. Just very quickly, if I may. This is the degradation that there was this is the 50s. Huge erosion problem, big overexploitation by cotton, tobacco. And after it was reforested, we don't really know how much has been recovered and what are the normal functioning, how the normal functioning of the system is. So clearly we need tools which are quantitative that couple the various processes, energy, water, biogeochemistry, to understand the state of the system and properly manage it. And so we're doing hydrology, we're doing soil formation studies, of course, large-scale landscape evolution type of analysis. And there are several, it's a whole network of them. These are just some preliminary results from a paper in review with Tony Parolari, my postdoc, which shows similar, these are data, these are theoretical models, but you see the same spiraling. And this is the soil plant system, similar predator-prey system. So once you perturb the system, the response is not necessarily monotonic, and that's key to understand. Now, I'm running out of time. I'll skip the next slide, which is just, I'm actually going to comment it very briefly. It's just when you harvest a forest, of course, you have these different responses depending on the biogeochemistry, carbon and nitrogen and so on. But I want to conclude just by summarizing three messages that hopefully I made clear. One is like, it's interesting, complex system where the space-time dynamic interaction of biotic and abiotic component is really driven by soil water availability. So hydrologists have a nice niche looking at this interdisciplinary and have lots to learn from different disciplines to put together a comprehensive view of these ecosystems. It's very much an open field of research, in my view, which has to combine laboratory field experiments with theoretical analysis. And it needs to be quantitative. It cannot be just, oh, let me see the linkages. I need to write. I need to get numbers to provide answers towards a more sustainable management of this ecosystem. Okay. I'll close here, otherwise we will do that like these guys. This is Xue Feng, my student, Giulia Vico, my postdoc. I'm, of course, just supervising them as we are coming back from a field experiment in Brazil. But thank you very much.
Before we take questions, I think you should go back to the mushroom ring slide, because I've never seen that before.
Yeah, it's very capturing. It's So I don't know exactly the story. So you're really, but so what is observed is that there is this. They're called, I think, fairy rings, and some people here may know much more than. But every year they grow. Like visually, it looks really like as if you are taking throwing a stone in a pond. Linear waves that propagate, and but clearly it's due to the fact that there is some. I don't know what, microbial interaction with the nutrients. And of course, this is I guess this is a soccer field or something. So it's very homogeneous, but you do see them, and every year they, I don't know what the equations are.
Yeah. Okay, questions? Yeah.
Very interesting talk. These spiraling patterns that you're getting in some of the data, it looks a lot like what Halling explored in biological systems with persistence back in the 70s, which was then used by May to look at multi-stability systems. And I'm wondering if you've explored your data for multi-stability.
Yes. So it's not just me, but colleagues, including Paolo Dudorico, a colleague at the University of Virginia. It's a very good observation. These feedbacks are very prone to at least bistability. One state being the degraded one. So if you go back to my sketch here, in some cases, you can really find, once the system goes into a perturbed state, a degraded state, like the Calhoun case that I showed here, it's very hard for the system to go back to the non-degraded state, because there are local feedbacks that lock the system there. So yes, but those don't typically take place just if you consider microbe substrate, but you need to see the whole soil system state. But yes, same thing with salinity. Once you lock your system into a saline state, like in Australia, once they cut the eucalyptus and the water table saline came to the surface, almost impossible then for eucalyptus to then grow again and draw the water table down, because the small seedlings just don't. So yes, plenty of these multiple states exist, and many of them need to be explored more in depth. Like, I don't know if my 8th system state has those. I was not able to do so far a full stability analysis. If you're volunteering to help, I'll be very, very glad to.
Howard.
Yes.
A very interesting talk, and on a hugely important problem. I think nutrients are one of the big challenges that we have. I'm really impressed at what you're trying to do, which is essentially to encapsulate, as I understand it, really complex processes in a fairly simple way, so that you can explore system properties. And I think that's really important, and of course we're looking for some insights from that. My understanding of the area of work is that we have this tremendous dichotomy between the complexity of the processes that we're working with and the very limited capability that we have in terms of data to support the analyses. So if we're interested in looking at nutrients at catchment scale, we're really struggling. And you can either use very simple methods, export coefficients, things like that, which have some strong foundation in empiricism, or else then you can go to models and which try to capture the main processes. And even relatively simple models then turn out to be very rather complex. And my experience of working with the sort of traditional family of conceptual models for nutrients is that actually they're very poorly determined. And they, although people think about them as perhaps being physically based, essentially they're expert systems, because you can actually play with them and get many, many modes of response out. So it's a question of your subjective prior choice of parameters. So you didn't talk about parameterization at all, and maybe you could talk about a little bit about the data support and the extent to which these models are identifiable,
Yes.
and the extent to which they can be applied.
Oh, that's a very good point, and it puts. But so maybe I can use an example to answer. I mean, completely agree that many times the data are. So I have two cases where I show data. One was just a very conceptual with the Duke Force data. But if I go back to the 2 points where I show the more serious data, one is here, and so I can definitely detect here, besides the noise, a curvature, which is a sign of a switch of one of the variable of the fluxes in the analysis. So it's a pretty compelling argument that can be used to then say, oh, there needs to be that flux. I cannot do to provide this plateau. I need to have where is that? Here. I need to have a flux going backwards. If I have a model that doesn't have immobilization, I cannot capture them. So I think that's the value of going simple, because I can really decide, rule out some, or have to take necessarily some of the processes. The other example I would like to cite is this one. So when I see oscillations like that, now I can have different nonlinear models that do that. Like the multiplicative model does a decent job, is the red one. There is another model better than ours, more nonlinear, that does a better job, but certainly I cannot use the linear model, because those fluctuations qualitatively are not present. So I don't want to criticize other models, but the models that use, and some famous models, are just based on a linear formulation. If they may do a very good job in some things, but if they will never be able to get those fluctuations. So I know there is still lots of room to.
Have you done any intercomparison with other models that might be more commonly used? Because I'm sort of conscious of the analogy with perhaps some of Jim Kirshner's work, where he's looked at frequency domain response of water quality, and essentially shown it's fairly inconsistent with the kind of modal response that you get out of most conceptual models. And I wonder if you could tell the same kind of story, really, that if you put together the kind of box models that people use,
yes,
you perhaps would find that they were not reproducing the modes of response that you're observing here.
Yeah, definitely. So Stefano, who is a very thorough, creative, but also very thorough researcher, had a review after this exactly to address that. So let's go to the literature. There are, I think he cataloged 260 biogeochemistry models from the, and we published that. So I'll pass you that it's a long table, and it goes by what is the number of degrees of freedom? Is it linear? Is it nonlinear? What are the parameters? And he really tried to classify that. And the interesting thing is that there is a clear gap in modeling and the scale at which you apply that model. So many times you just take an off-the-shelf model which is made for, I don't know, daily scale, plot scale, and you see it applied at the watershed scale at, I don't know, half an hour, where the processes are just not there. So I'll pass you that review.
Okay, we're just after 5. I think we should draw it to a close. For those that had their hands up and would like to ask a question, please see Amilcare at the bar afterwards. We'll be making a move to Alexander's. I'd like to thank Amilcare for a stimulating talk and a great day of discussions.
Oh,
Thanks
thank
very
you.
much.
No, it was my pleasure.